Have you ever wondered why two houses look different even though they are made of the same raw materials, bricks, cement, mortar, etc.? It is only because of the difference in the architectural plan of the two houses that they appear so different. You must be wondering why we are talking about buildings and architecture in a biology class. Okay, let me tell you why. Although all houses are made up of bricks and cement, but still they look different. Similarly, all of us are made up of the same basic material, the cells, having deoxyribonucleic acid, that is DNA, but still we look so different. Actually, expression of characters is governed by genes located on the DNA, so it is our DNA which makes us different from each other.
Students, in biology classes, you often hear that DNA is our hereditary material. In simple words, DNA contains instructions for making proteins within the cell which help to create distinctive features in an individual's body. What are these instructions? Basically, genes are instructions that control everything which happens inside a cell. In our lesson today, we will mainly discuss the structures of DNA and RNA. While discussing their structure, we will also come to know the differences between a nucleoside and a nucleotide that are the main constituents of DNA and RNA. Students, DNA and RNA are two types of nucleic acids found in living systems. DNA acts as a genetic material in most living organisms. RNA also acts as a genetic material, but only in some viruses. ਤੁਹਾਡਾ DNA ਤੁਹਾਡੇ ਫਿੰਗਰਪ੍ਰਿੰਟ ਵਾਂਗ ਹੀ ਹੁੰਦਾ ਹੈ, ਜੋ ਕਿ ਹਰ ਵਿਅਕਤੀ ਦਾ ਵੱਖਰਾ ਵੱਖਰਾ ਹੁੰਦਾ ਹੈ, ਪਰ ਇਹ ਵੇਰੀਏਸ਼ਨ ਬਹੁਤ ਜ਼ਿਆਦਾ ਨਹੀਂ ਹੁੰਦੀ। ਤੁਹਾਨੂੰ ਇਹ ਜਾਣ ਕੇ ਬਹੁਤ ਹੈਰਾਨੀ ਹੋਵੇਗੀ ਕਿ ਤੁਹਾਡੇ ਕੁਝ ਜੀਨਸ ਬੈਕਟੀਰੀਆ, ਕੈਰਟਸ, ਪਾਈਨ ਟਰੀ ਅਤੇ ਇੱਥੋਂ ਤੱਕ ਕਿ ਬੈਟਸ ਨਾਲ ਵੀ ਮਿਲਦੇ ਜੁਲਦੇ ਹਨ। ਪਰ ਇਹ ਜੀਨ ਕੀ ਹੁੰਦਾ ਹੈ? A gene is a distinct portion of DNA, or we can say genes are made up of DNA. Genes are segments of DNA passed from parents to offspring. ਇਹੀ ਉਹ segments ਹਨ ਜੋ ਕਿ hereditary information ਮਾਂ ਪਿਓ ਤੋਂ ਬੱਚਿਆਂ ਤੱਕ ਪਹੁੰਚਾਉਂਦੇ ਹਨ। Hereditary information is that information which is passed on from parents to their children. Let us recall where this DNA is present in our body and how it looks like.
We know that nucleus contains chromatin material, and just before cell division, this chromatin material gets condensed to form chromosomes. Here you can see a cell prior to cell division. In the nucleus of this cell, the chromatin material condenses to form chromosomes. If we look at the detailed structure of a chromosome, these are thread-like structures. These thread-like structures consist of DNA and some proteins. Actually, DNA has to be present in a very compact form to fit into the small nucleus.
Do you know how the information present on DNA within the nucleus reach the cytoplasm and the protein synthesizing sites? Yes, it is through ribonucleic acid, that is RNA. The information on the DNA is transcribed into RNA, which moves out of the nucleus into the cytoplasm, where it gets translated into proteins by the protein synthesis machinery. So, to understand complicated processes of transcription and translation, it is important to learn the structures of DNA and RNA. In today's class, we will study about the structures of DNA and RNA in detail. ਮੈਂਡਲ ਵੱਲੋਂ ਤਜਵੀਜ਼ ਕੀਤੇ ਇਨਹੈਰੀਟੈਂਸ ਲਾਅ ਤੋਂ ਬਾਅਦ ਬੇਸਿਕ ਜੈਨੇਟਿਕ ਮਟੀਰੀਅਲ DNA ਡੀਆਕਸੀਰਾਈਬੋਨਿਊਕਲੀਇਕ ਐਸਿਡ ਦੀ ਬਣਤਰ ਨੂੰ ਸਮਝਣ ਲਈ ਲਗਭਗ 100 ਸਾਲ ਲੱਗ ਗਏ। ਪਾਵੇ ਫਰੈਡਰਿਕ ਮਾਈਸ਼ਰ ਨੇ 1869 ਵਿੱਚ ਨਿਊਕਲੀਅਸ ਵਿੱਚ DNA ਦੀ ਮੌਜੂਦਗੀ ਇੱਕ ਤੇਜ਼ਾਬੀ ਸਬਸਟੈਂਸ ਵਜੋਂ ਕੀਤੀ, ਪਰ ਇੱਕ ਲੌਂਗ ਪੋਲੀਮਰ ਨੂੰ ਵੱਖ ਕਰਨ ਵਿੱਚ ਤਕਨੀਕੀ ਕਮੀ ਦੇ ਕਾਰਨ DNA ਦੀ ਬਣਤਰ ਨੂੰ ਲੰਮੇ ਸਮੇਂ ਤੱਕ ਨਹੀਂ ਸਮਝਿਆ ਜਾ ਸਕਿਆ। In 1953, James Watson and Francis Crick proposed the double helical structure of DNA. Their interpretation of structure of DNA was based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. Let us explore the components of DNA in detail.
The repeating unit. Each unit of DNA is composed of a pentose sugar, a phosphate group, and a nitrogenous base. Collectively, they are known as a nucleotide. So, the repeating unit of DNA is a nucleotide. Backbone of DNA is formed by repeated pattern of pentose sugar and phosphate. As its name indicates, DNA contains deoxyribose sugar. Deoxyribose sugar is formed by the loss of an oxygen atom from the ribose sugar. The carbon atoms of the sugar molecule are numbers with a dash above them to distinguish them from the carbon atoms of the nitrogenous bases, and the numbers are read as 1 prime, 2 prime, and so on. The second component of the repeating units of the backbone is the phosphate group. The phosphate group links with the sugar by replacing the hydroxyl group present on the 5th prime carbon of one sugar and 3rd prime carbon of adjacent sugar by phosphodiester bonds. The third component of the repeating monomeric unit of a nucleic acid molecule is the nitrogenous base. There are 2 types of nitrogenous bases, Purines and Pyrimidines. In case of DNA, adenine and guanine are the purines, and cytosine and thymine are the pyrimidines. Adenine and guanine are bigger because they have 2 rings each. Cytosine and thymine have only 1 ring each. These four bases are represented by the first letters of the base name. A for adenine, G for guanine, C for cytosine, and T for thymine. A nitrogenous base is linked to the pentose sugar through an N-glycosidic linkage. The monomeric unit, composed of a phosphate group, a pentose sugar, and a nitrogenous base, is together known as nucleotide. The four types of nucleotides are deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. The pentose sugar, along with the base, is called nucleoside. The four types of nucleosides in DNA are named as deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine.
ਇਹ ਤਾਂ ਸੀ DNA ਦੇ ਅੰਦਰ present chemical components ਬਾਰੇ ਜਾਣਕਾਰੀ। Before moving further, ਚਲੋ ਚੈੱਕ ਕਰ ਲਈਏ ਕਿ DNA ਦੀ chemical composition ਬਾਰੇ ਤੁਸੀਂ ਕੀ ਸਮਝਿਆ ਹੈ। ਪਹਿਲਾ question ਕੁਝ ਇਸ ਤਰ੍ਹਾਂ ਹੈ। A nucleotide is composed of, the options are, a nitrogenous base and a phosphate group, a pentose sugar and a phosphate group, a pentose sugar, a phosphate group, and a nitrogenous base. A pentose sugar and a nitrogenous base. Answer. You got it right. The correct answer is this option. A nucleotide is composed of a pentose sugar, a phosphate group, and a nitrogenous base. Next question. There are two types of nitrogenous bases, dash and dash, present in DNA. Answer. There are two types of nitrogenous bases, Purines and Pyrimidines, are present in DNA. Next question. A nitrogenous base is linked to the pentose sugar through an dash linkage. Answer. A nitrogenous base is linked to the pentose sugar through an N-glycosidic linkage. ਤੁਸੀਂ ਇੱਕ ਨਿਊਕਲੀਓਟਾਈਡ ਦੇ ਸਟਰਕਚਰ ਨੂੰ ਸਮਝ ਲਿਆ ਹੈ। ਚਲੋ ਹੁਣ ਸਮਝੀਏ ਕਿ ਇਹ ਛੋਟੀ ਯੂਨਿਟਸ ਮਿਲ ਕੇ ਕਿਵੇਂ DNA ਦੀ ਲੰਬੀ ਚੇਨ ਬਣਾ ਲੈਂਦੇ ਹਨ। Let us move further to explore more about the structure of DNA.
The polynucleotide chain. A DNA strand is simply a string of nucleotides joined together. The phosphate group connected to 5th prime carbon of its own nucleotide links to the 3rd prime carbon atom on the sugar of the adjacent nucleotide. The linkage between two nucleotides is called as 3' to 5' Phosphodiester linkage. In the process, a molecule of water is lost and a dinucleotide is formed. Similarly, more nucleotides are added and thus a polynucleotide chain is formed. This chain has a free phosphate group at the 5th prime carbon of the first nucleotide which is referred to as the 5' end, while the other end has a 3rd prime hydroxyl group of the last nucleotide added. This end is referred to as 3' end of the polynucleotide. The sugar and phosphate groups of the polynucleotide form the backbone, while the nitrogenous bases project from the backbone.
DNA ਦਾ ਡਬਲ ਹੈਲੀਕਲ ਸਟਰਕਚਰ ਇਸ ਦਾ ਨਾਂ ਤਾਂ ਤੁਸੀਂ ਕਈ ਵਾਰ ਸੁਣਿਆ ਹੋਵੇਗਾ। ਚਲੋ ਪਹਿਲੇ ਡਿਸਕਸ ਕਰੀਏ ਕਿ ਇਸ ਦੀ ਫਾਰਮੇਸ਼ਨ ਕਿਵੇਂ ਹੁੰਦੀ ਹੈ। ਇਸ ਤੋਂ ਬਾਅਦ ਇਸ ਨੂੰ ਵਿਜ਼ੂਅਲਸ ਦੀ ਸਹਾਇਤਾ ਨਾਲ ਸਮਝਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰਾਂਗੇ। ਪਰ ਸਭ ਤੋਂ ਪਹਿਲਾਂ एरविन चारगाफ द्वारा नाइट्रोजनस बेसिस बारे दिते गए कुछ फैक्ट्स नु समझ लइए। Erwin Chargaff and his colleagues analyzed the base composition of a large variety of DNA and found that the pairing of bases is very unique. Adenine of one chain is always paired with the thymine of the second chain, and a guanine of one chain is always paired with the cytosine of the other one. It was also observed that in a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equal to 1. चलो हुण वॉटसन and Crick दे double helical structure नु समझ लइए। According to Watson and Crick, the two polynucleotide chains made from deoxyribonucleotides are joined together by pairing of bases through hydrogen bonds. The two strands are said to be complementary to each other so that each strand can act as a template for the synthesis of the other strand, and the two DNA molecules produced will be identical to the parent strand. ਮੈਨੂੰ ਲੱਗਦਾ ਹੈ ਕਿ ਵਿਜ਼ੂਅਲ ਰਾਹੀਂ ਹੀ ਅਸੀਂ ਇਸ ਨੂੰ ਚੰਗੀ ਤਰ੍ਹਾਂ ਸਮਝ ਸਕਦੇ ਹਾਂ।
In a double helical structure given by Watson and Crick, the two polynucleotide chains are helically wound to each other by pairing of bases through hydrogen bonds. The specificity in pairing of bases is a result of formation of very effective hydrogen bonds between adenine and thymine and guanine and cytosine. Salient features of Watson and Crick's double helical DNA. Each DNA double helix is made from two right-handed helical polynucleotide chains. The two chains are opposite in polarity. The two chains are coiled around the same axis. The bases always project towards the inside of the helix. They form pairs with bases on the opposite strand in such a way that a pyrimidine base attached to one strand is always paired with a purine base attached to the opposite strand. Base pairing is facilitated by formation of hydrogen bonds between complementary bases. The distance between two successive base pairs is approximately 0.34 nanometer. Each turn of the double helix covers a distance of 34 nanometer, thus containing 10 sets of base pairs. Each base pair is turned 36 degrees from the preceding pair, so that there are 10 base pairs in a complete turn of 360 degrees. The plane of one base pair stacks over the other in a double helix. This feature provides extra stability to the helical structure.
We know that each chromosome is made up of long strands of DNA. You must be wondering how these long strands of DNA are packaged in such a small nucleus. To accommodate itself in the nucleus, DNA has to be highly organized. In prokaryotes, as you know, there is no defined nucleus, but still, DNA does not remain scattered in the cytoplasm. It remains in the form of loop, known as nucleoid. It is possible because negatively charged DNA is stabilized by the positively charged histones. Thus, in prokaryotes, DNA remains organized in the form of large loops held by proteins. In eukaryotes, this organization is much more complex. Let us see how DNA remains tightly packed in a small nucleus of eukaryotes.
DNA packaging in eukaryotes. There is a set of positively charged basic proteins called histones. DNA is wrapped around a histone octamer to form a structure called nucleosome. Histones are basic proteins which have positive charge on them. This positive charge is due to the presence of amino acids, lysine and arginine, in excess amount. DNA takes 2.5 turns around each histone octamer. The coiling of DNA around the nucleosome shortens the length of DNA fivefold. Each nucleosome has a diameter of 125 Armstrong and contains about 200 base pairs. The nucleosome is the first stage of DNA contraction. It is further compacted by grouping of nucleosomes. Two nucleosomes are linked to each other by a small section of interconnecting spacer DNA, to which is bound another histone protein, H1. Under an electron microscope, the DNA looks like a bead on a string. The beads are nucleosomes and the string is the spacer DNA.
DNA is further compacted by additional sets of proteins that pack the chromatin into chromatin fibers. These proteins are collectively known as non-histone proteins. The chromatin fibers are, however, not uniformly packaged. When viewed under microscope, Some parts of the chromatin appear to be darkly stained due to their dense packing, while other parts are loosely packed and therefore stain light. The darkly stained chromatin is called heterochromatin and is said to be transcriptionally inactive, while lightly stained chromatin is called as euchromatin and is said to be transcriptionally active. ਚਲੋ DNA ਨੂੰ ਤਾਂ ਅਸੀਂ ਚੰਗੀ ਤਰ੍ਹਾਂ ਸਮਝ ਲਿਆ ਹੈ। ਹੁਣ RNA ਦੀ ਗੱਲ ਕਰੀਏ। RNA is a nucleic acid polymer that plays an important role in the processes that translates genetic information from DNA into proteins. Unlike DNA, which is mainly found in the nucleus, RNA is found in all major cell fractions. RNA is a polymer with a ribose sugar and phosphate backbone. Like DNA, RNA is also composed of two types of nitrogenous bases, purines and pyrimidines. The only difference from DNA is that the pyrimidine thymine is replaced by uracil in RNA. RNA mostly exists as a single-stranded molecule and has a short chain of nucleotides. Let us see three different types of RNAs.
This is mRNA, means messenger RNA. It carries information to construct a protein. It is transcribed from DNA and taken to ribosomes. Ribosomes read this mRNA to link amino acids to form a specific sequence. This is tRNA, means transfer RNA. It has a coding section and an amino acid carrying section. The code identifies which amino acid is carried so that the proper amino acids are used by ribosomes during protein synthesis. So, tRNA transfers specific amino acids to the growing chain of polypeptide during translation. Here is rRNA, means ribosomal RNA. It is also transcribed from DNA but is not a code carrier. It binds protein and mRNA to provide the site for protein synthesis.
This brings us to the end of the lesson. Let us take up some questions just to check our understanding of the topic. The first question is, deoxyadenosine, deoxycytidine, deoxyguanosine and deoxythymidine are examples of ribonucleosides, ribonucleotides, deoxyribonucleosides or deoxyribonucleotides. Answer. You got it right. Deoxyadenosine, deoxycytidine, deoxyguanosine and deoxythymidine are examples of deoxyribonucleosides. The next question is, what will the complementary sequence of bases that will pair with AGCC? The options are TCGG or TCTT or TAGG or TGAA. The answer is, you guessed it right. The correct answer is the first option, TCGG. You will be able to answer such questions only if you know about the findings regarding base pairing given by Erwin Chargaff. चलो एक होर इसी तरह दे सवाल दा जवाब दियो. Which of the following base pairs is correct? Adenine guanine or adenine thymine or guanine thymine or adenine cytosine? The answer is adenine thymine. Hope I have made a successful attempt to clear all your concepts regarding structure of DNA and RNA. Thanks for your cooperation and attention. Looking forward to the next class. See you then. Thanks.